Hi everyone. So for the last part of Module 2, Part 2.3, we will be focusing on microbiomes outside of the gut, how different body sites have different microbial profiles, and even how a single body site may have different microbial compositions. So as a start, let's look at the oral microbiome. Advances in molecular studies have shown that, for example, periodontal disease, which affects the soft tissues and bone that support our teeth, and caries, which is a unique infection of the dental hard tissues, may result from a perturbation among relatively minor constituents in local microbial communities, resulting in dysbiosis rather than from a single pathogen as previously thought. Not only does the oral microbiome differ in health and disease states, but it also differs in composition from one intraoral site to another. The oral cavity includes several distinct microbial habitats, such as periodontal pockets and the surface of teeth and cheeks. The tongue has been identified as the most populated niche and has a significant impact on other regions in the oral cavity. In addition to the different oral diseases such as caries and periodontitis, the oral microbiome has also been associated with systemic diseases. For example, oral microbial dysbiosis has also been associated with gastrointestinal system diseases like inflammatory bowel disease, liver cirrhosis, pancreatic cancer, nervous system diseases like Alzheimer's disease, endocrine system diseases like diabetes, adverse pregnancy outcomes, obesity, and polycystic ovaries syndrome, immune system diseases like rheumatoid arthritis and HIV infection, as well as cardiovascular system diseases. The skin, on the other hand, is another body site with diverse topographical microbial distribution, which is highly dependent on the microenvironment of the sampled site. For example, to your microbes, the differences between your heel and your toes may be worlds apart. We'll focus on the respiratory microbiome. In 2013, the majority of deaths in children were resultant of infectious disease. Of these infectious diseases, pneumonia remains the leading cause of death among children under 5, with the majority of its victims being under the age of 2 years. The conventionally understood mechanism of pathogenesis of bacterial pneumonia is that the lungs are sterile and that a suitably large inoculum of a pathogenic species enters the lower respiratory tract from the upper respiratory tract and overwhelms host defenses. However, advances in techniques used to study the microbiota of the lungs have now shown that the lungs are not sterile and contain diverse and dynamic communities of microbes. Pathogenic species causing acute infections are also detected in the microbiota of healthy people. The respiratory tract is a single ecosystem extending from the nares to the alveoli, comprising a continuous and continuously varying microbial profile. Changes in these profiles might result in lung diseases such as pneumonia. In Part 2.2 of this module, breast milk was introduced as a mode of infant feeding, which has been associated with a number of health benefits for the infant. Breast milk in itself contains a complex community of bacteria that may help seed the infant gut microbiota. However, the composition and determinants of milk microbiota are poorly understood. Musavi and colleagues investigated the milk microbiome of 393 nursing mothers in order to determine potential determinants of breast milk. Using causal modeling, their results suggest that multiple maternal, infant, and environmental factors interactively influence milk microbiota composition. Most strikingly, they found that indirect breastfeeding and pump expression were consistently associated with milk microbiota composition, highlighting the importance of breastfeeding practices. These data support the retrograde inoculation hypothesis, whereby the infant oral cavity impacts the milk microbiota. Interestingly, gender differences were also identified, as well as potential associations between microbiota and other milk components. One of the interesting findings from vaginal microbiome studies is that more diverse does not always mean healthy. A number of studies, especially studies focusing on the gut, have reported that less diverse profiles are associated with disease states. In vaginal microbiota, this is just the opposite. But before we look at some of these interesting findings, I first want to talk to you about the term diversity. So what do we mean when we talk about microbial diversity? A diversity index is a quantitative measure that reflects how many different types of organisms are present in an environment and how are they distributed. The richer the environment sampled, which translates to more different organisms from a single environment, and the more evenly the sampled environment, which translates to more similar distribution of different organisms, the more diverse the environment sampled. Srinivasan and colleagues showed that women with bacterial vaginosis had more diverse bacterial communities, as you can see by this beautiful figure, whilst in the absence of bacterial vaginosis, the vaginal microbiota was dominated by only 2 lactobacillus species. So for this part of Module 2, we have looked at different body sites and how their bacterial communities differ between and even within sites. Throughout this module, I've highlighted only a few interesting findings from studies using the 16S rRNA gene sequencing technology to study collections of microorganisms present at different sites. However, the term microbiome has been used very loosely in my presentations up to this point. And up until now, we haven't actually focused on what exactly is meant by the term microbiome. The term microbiome is defined as the genetic material or collective genomes of all the microbes, which include, for example, bacteria, archaea, viruses, and fungi in a particular environment. Assemblages of these organisms belonging to different kingdoms are respectively referred to as bacteriomes, archaeomes, viromes, and microbiomes, which all constitute the umbrella term microbiome. For the purpose of this course, we will primarily be focusing on the 16S rRNA gene, the most commonly used bacterial target, and high-throughput sequencing, also referred to as next-generation sequencing, which allows for simultaneous sequencing of DNA from diverse environments containing various bacterial species during a single sequencing reaction. The use of next-generation sequencing technology has increased the size of the 16S rRNA gene sequencing database at an impressive speed, and by just typing in 16S sequencing and health in the PubMed search bar, it is fascinating to see how much this fairly recent technology has contributed to better understand the role of our bacterial communities in health and disease states. So for Module 3, I will be referring to microbiomes studied by targeting the 16S rRNA gene, specifically as bacteriomes or bacterial profiles. In summary, microbial communities exist outside the human gut. A number of studies have shown that microbial communities detected at different body sites are associated with health and disease states. Microbial communities are different for different body sites, and we even find different communities within different topographies of the same body site. The term microbial diversity is defined as the number of different organisms and how evenly they're distributed within an environment. The term microbiome is an umbrella term for a number of microscopic organisms detected in any given environment. These include bacteria. 16S rRNA high-throughput sequencing is used to study the bacteriome or bacterial profiles within any given environment. So that's the end of my introduction to the human microbiome and why it's important to study our bacterial communities using 16S rRNA gene sequencing. I hope that Module 2 has helped you to better understand the term microbiome and also emphasized that we need to use this term and related terms such as bacteriomes, viromes, and microbiomes correctly. The module also introduced current hypotheses around the ways we may be acquiring our microbes and highlighted that our microbial profiles may be influenced by a number of factors, especially early life exposures. Finally, our microbiomes consist of more than just the gut microbiome, and microbial communities outside the gut are also impacting on our health and disease states.